Morning.
Good morning.
Morning.
First, any questions or concerns?
I was just going to ask, did you get my email?
When did you send it? Was it, I received two like last night. Was that them, Quentin?
Let me see in my sent. Yeah, December 6th. Yeah.
I got them. Have I read them? No. What was, what did they say?
It was about what we talked about last class.
Okay. Can I respond? Can we talk about it after class?
Yeah.
All right, let's talk about it after class. Other questions? Other questions? You know what, Quentin? I don't think I've received one from you. Actually, no, I did. Yes, I can do it. Quentin, just know that I'm saying yes. Okay.
Okay.
Okay. Other questions? All right, your third case study is posted. And I apologize for slamming you with case studies right now. Kind of. The reason I'm also doing it is before I gave you a case study, I wanted to make sure that you had some real knowledge about anatomy and physiology. And if this semester has been anything like it has been for me, it's kind of been a blur. It goes by really, really, really fast. And there's a lot of information. And I kind of what's really funny, by the end of the semester, my only thought sometimes is we made it and nobody died. And that's what matters. And that may sound really funny, but it is meant to be a little funny. But and for some, well, at least one of you, that may not be so funny. But you've made it. And the reason I gave you all these case studies was because I liked you to be able to apply what you're learning. And so there is one more. And do me a favor, read through it between now and Wednesday. Read through it. And I want you, what am I doing? Read through it and let me know what you have questions about. Because if you have questions about it, someone else does too. And if we have time on Wednesday, I will read through it with you. But we have to finish chapter 11 today. And we have to start we have to finish chapter 11 today, and we have to do the lab that we have planned on Wednesday. And that will be the end of the semester. However, what I want is when you look at these case studies, bear with me. Actually, I'm going to open it really quickly. Nice thing about this class, because I teach both the lecture and the lab, is What if we don't get to something today, I can squish it in lab too. Whether you like that or not, I don't know, but I like that I can do that. Looking at this lab, there's two different case studies going on here. There's a case study in muscle contraction, which we talked a little bit about lab last week. We'll spend more time talking about it this week. And initially what these are doing is just asking you a bunch of definitions and matching them up. There's only one on here that you may not be familiar with, and I realize your book doesn't really talk about it. And let me see if I can tell you the answer right now. So you're an intern working in the Atlanta, Georgia office of Dr. Priya Wayne. Dr. Wayne is a specialist in rare neuromuscular and musculoskeletal disorders. You've been working with Dr. Wayne for the last week, and due to this experience, you've gained a great deal of knowledge about the human body and muscle physiology. You're also a college student working with Dr. Wayne, and she has allowed you to gain knowledge, firsthand experience about some of the material you're learning in your human physiology course. But last week you turned in an assignment comparing the disorders of the neuromuscular junction. You learned all about several issues, including myasthenia gravis, SARA-Curare, botulism, and Eaton-Lambert syndrome. We'll talk about some of these today. Talk about myasthenia gravis, and curare. Maybe we won't talk about curare, but we'll talk about botulism. Today, a group of high school students is coming for a tour, and Dr. Wayne has asked you to prepare for some information about muscles to present to the students. Specifically, Dr. Wayne has asked you to discuss the neuromuscular junction, muscle contraction, and explain some of the issues that can occur when signaling between neurons and muscles doesn't go as planned. We talked a little bit about this last week in lab. We'll review it again today also. So this is a word bank, and what you're being asked is to match up with the different words. And the answer for the ryanodine receptor is number 6. It's a receptor that's located on the sarcoplasmic reticulum. And once opened, it allows calcium to flow from the sarcoplasmic reticulum into the sarcoplasm. Did you hear what I said? Let me just repeat that for you. The number 6 is the ryanodine receptor. We just talk about the fact that this is actually the receptor to which the action potential will get opened. That's going to release the calcium into the action potential will open, which will release the calcium from the sarcoplasmic reticulum. That's the receptor. Remember, we always have receptors on cells or organelles. That's a receptor. Read through the rest of this between today and Thursday. see if you can't answer a lot of these questions. This last one you should be able to read. Briefly describe the role of the autonomic nervous system in human physiology. What are the two divisions of this system? Well, what are the two divisions? It's the sympathetic and the parasympathetic nervous system. What is the role of it? Its major role is in homeostasis of the body and reflexes. It's involuntarily controlled, and it helps maintain homeostasis of the body. And if you go to chapter 15, before you answer any of these questions, what I want you to do first is look in the PowerPoints. When did we talk about the autonomic nervous system? It was chapter 15. Bear with me just for a second here. And another thing that I plan to do between now and next semester is definitely arrange the PowerPoints differently. There's always things every semester that I think I will definitely change and improve on before next semester. And it's that way every semester for me. But the autonomic nervous system and visceral reflexes, what are the main divisions? The sympathetic and the parasympathetic. If you want to include the enteric, go ahead and include the enteric in there. It maintains homeostasis. The role of it is to maintain homeostasis. It does it by regulating the activities of the smooth muscle, cardiac muscle, and glands. That's your answer. Do you understand why I'm telling you when you're looking at these case studies, go to the PowerPoints first? If you can't find it in the PowerPoints, go to your book second. But if you want my help, send me a text or an email and tell me, can you help me with this question? And I'll tell you where in the PowerPoint to look for it. Because I'm specifically directing these questions towards things we've talked about. Are there any questions? So your third one, 1, 2, and 3 is right here. And like the other two, it will be 30 points. It will be worth 30 points. Your third exam will open today at 1:30. And it's going to cover the rest of the chapters. So it's going to cover 11, 15, and 16. Let's jump into this chapter and finish this chapter on muscle contraction. We were on 28. So I lose the top of my screen, which tells me where I'm at. So remember the structure, the overall structure of muscle. We have overlapping thin and thick bands. Actually, I'm going to back up just a little bit. Actually, maybe I'm not going to, because I don't think it'll help. We have overlapping thin and thick bands. The thin bands are actin. The thick bands are myosin. The myosin is attached to the Z disc via the titin protein, which runs all the way through the thick filament. From one Z disc to the next Z disc is one sarcomere, which is a functional unit of muscle. The H band is where there's only a thin muscle filament. Excuse me, only a thick muscle filament. The A bands runs the entire length of the thick filament. And it's really only looking at the thick filament. The H band is the lighter center at the middle of the A band. Remember A, if you remember it this way, it's an alternating I and A bands that are creating the striations in muscle. The H band is where that is the middle of the A band, and it looks a little lighter because it's only the unexposed thick filament. The I bands are only the thin filaments. The Z line runs through the middle of the I bands. The M line runs through the middle of the A bands. Questions? This is going to be really important in a few minutes here. Understanding this arrangement. And I know that sometimes it helps to write this out a few times and draw this out, the overlapping of it. Because what muscle contraction is it's a sliding of these filaments on top of one another. The myosin pulls the actin to the center through its binding to the actin and pulling it forward, pulling it towards the center. And as a result, what happens is the two Z bands are getting closer together. The two Z discs are getting closer together. And so the whole length of the sarcomere shortens. Well, the muscle fibril is one sarcomere attached to another sarcomere, attached to another sarcomere, attached to another sarcomere, and so on. As one Z, as two Z discs get closer together, one sarcomere shortens. The next sarcomere also shortens, the next sarcomere also shortens, the next sarcomere also shortens. The result is that the whole fibril is going to shorten in length as they're all shortening. That is the way muscle contraction occurs. Actually, this a little differently real quick. So here we're gonna make it very. I'm gonna try to draw it reasonably well here. See, actually, let's do that a little smaller. Here's a Z disc. 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 And in the center of them, we have the overlapping, the thin filaments, like so. And then we have the thick filaments. Here's the thick filaments. Here's the thick filaments. Okay, so here's a myofibril. It would continue on this way. It would continue on going towards the right and the left. But this is when it's relaxed. When it contracts, this sarcomere shortens in length. 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 So this is it relaxed. This is it when it contracts. So myofibril relaxed, myofibril contract. And I'm sorry, I apologize. I do the best I can with this whiteboard, but I have to say writing with your mouse is sometimes like writing like you were in kindergarten again. But let me. I'm looking at. It worked out a little bit, but I'm still pretty stringy. I've always been kind of a string bean. But here's my biceps right there. Here it's full, it's relaxed, full extended out, full extended out. When it contracts, the myofibrils in it, whole length here, are going to get shorter. If you look down, right about right here, right about right there, there's a tendon. That's the tendon of the biceps brachii, and it attaches on the radius down here. We'll spend a little bit of time talking about that on Thursday, how that structure is. But right here, it's in full length. It's relaxed. When it brings my radius up, it has shortened in length. So one of the myofibrils, or many of the myofibrils in the biceps brachii here, have all of a sudden gone from being this long to being this long. Because all of the sarcomeres within a given myofibril are all shortening. And when they're all shortening in length and they're attached to one another, the result is that the whole muscle is going to shorten. Questions? Is it clear? Someone tell me yes or no.
Yeah.
Okay. So what we're going to talk about almost to the very end here is essentially what I just said in with a little bit more detail. But it's the same process. So the Z discs are that make up the ends of the sarcomeres, and they're made up mostly of a protein called alpha actinin. The M line is made up of accessory proteins, which include myomessin, C proteins, creatine kinase, and they're attached to a protein called titin. Remember, titin is this big, thick filament here, or this big elastic filament. You going to bed? I was like, you can't take her. She's my teaching assistant. Yeah. Okay. Nebulin is an additional structural protein that wraps around the entire length of the Z filament, of the thin filament, and anchors that thin filament to the Z disc. So it would be similar in its function for the thin filaments as tightness for the thick filaments. It's just a reinforcing protein. Dystrophin is clinically the most important non-contractile protein in muscle contraction. So it's the most important structural protein clinically. And we'll see why here in a second. It links actin in the outermost myofilaments, so the outermost filament, to the membrane proteins that link to the endomysium, meaning the outer connective tissue of the muscle. It transfers forces of muscle contraction to the connective tissue out here. It helps to transmit tension. Muscle contracts, it helps transmit contention, tension. So it's, remember how it's attached here. It's linked from the most outermost thin filaments to the membrane proteins that are linked to the overlying endomysium, which is the top layer of the muscle connective tissue. You remember that fascia? This is one of those fascias, the endomysium. So why do we care about this protein? Why is it clinically so important? Because it's what is genetically mutated in Duchenne's muscular dystrophy. The gene that encodes without dystrophin, there's no reinforcing effect in being able to transmit tension. Or how this works, so it's right here, how it works, attaching the thin filaments to the top most outer plasma membrane. The result is that the sarcolemma, here's your sarcolemma right here. Your sarcolemma is going to tear easily during muscle contraction. Remember how you're built, you're born with all of the muscle fibers you're ever going to have. If they get damaged and you run out of satellite cells, which are involved in helping repair some of them, they're really important. And they're more important during hypertrophy, which we'll talk again about here in a little while. But satellite cells, if you run out of being able to build ability to use satellite cells to repair this, and you don't have any dystrophin, the result is that the muscle fibers rupture and die. You just can't, the muscles, it just lice. So that is why most individuals with Duchenne's muscular dystrophy are born with muscles that normally contract and relax. However, over time, they gradually lose all their muscle fibers, or the majority of their muscle fibers, to the point of where they die, mainly a lot of times from suffocation, because the diaphragm is a skeletal muscle also, and it has to be involved in contracting and relaxing. Questions? So striations are alternating A bands and I bands. Here's your I band, here's your A band, here's your I band, here's your A band. Here's your M line, here's your H band. Here's your Z disc. So there's about 10,000 sarcomeres that are banded together that span the length of the myel muscle fiber and give the appearance of the striations. So picture there's about 10,000 of these, each one of these. So the sarcomeres extend from Z disc to Z disc, and they are the functional contractile unit of the myofibril. The Z discs attach neighboring sarcomeres and separate one sarcomere from the next. These Z discs provide the anchorage for the thick filaments, the titin and the titin, the elastic filaments. The A band, which is the reason we call them the A bands versus the I bands, is A stands for what's called anesiotropic, which is indicative of the way that light reflects reflects the way the bands reflect the polarized light. Just know that they're dark. This is where thick filaments overlap with thin filaments. The H zone is not as dark, and it's in the middle of the H bands. And are only the thick filaments only. The M line is the middle of the H zone. Whether we call it a zone or a band, it doesn't matter. Just recognize that that's what it is. We'll say that it's synonymous here. Called an H zone here, called an H band up here. And I think both of these pictures came from your book, if I remember right. The I band stands for isotropic, so it's the same way. It's indicative of the way the light reflects the polarized light. The band reflects the polarized light. It contains the thin filaments and no thick filaments. The Z disc bisects the I band. I'm not going to go through these. These are just some review slides. Looking at a review of the structural organization. Stimulus of muscle contraction. Skeletal muscle never contracts unless it's stimulated by a nerve. If nerve connections are severed or poisoned, the muscle is paralyzed. If you remember from one of the first slides of this chapter, it talked about how in smooth muscle, if there's paralysis, it becomes hyperexcitable. In skeletal muscle, if the nerve detaches, it becomes paralyzed and undergoes what's called denervation atrophy, which means that the muscle shrinks. The muscle fibers start to shrink in size. A motor unit is one nerve fiber and all of the muscle fibers innervated by it. So one motor unit is one nerve fiber and all of the fibers innervated by that fiber. That's the definition. Muscle fibers of one unit can be dispersed throughout the muscle and they contract in unison simultaneously. They contract fully or not at all. It's that all or none law. If you remember, we talked about action potentials. Muscle, just like neurons, are capable of creating action potentials. And just like we saw with action potentials in neurons, in muscles, it's also an all or none. They can produce a weak contraction over a wider area. They have the ability to sustain long-term contraction as motor units take turns contracting. One motor unit is contracting now. Another motor unit will start to contract in a moment. Within your biceps, one motor unit contracts now. Another one is going to contract in a second. While the one that was just contracted relaxes. And then another motor unit is going to contract. Which altogether, because they're taking turns, allows for a sustained long-term contraction. Effective contraction usually requires that several muscle units are contracting at once. But you have lots of motor units in there. And so several can be contracting while others are going to contract next. When a motor neuron fires an action potential, how many skeletal muscle cells in that unit contract?
Wouldn't it be all of them?
Erin, why do you say that?
Because it's in the motor unit, it's all of the muscle fibers that the nerve fiber innervates.
You are 100% correct. Yes. And your reasoning is right too. So all of the fibers in there are going to contract. We have small motor units versus large motor units. Small motor units have a fine degree of control. There's 3 to 6 muscle fibers per neuron. These are your ones that are involved in when you're learning how to write out the alphabet. You're working on training these muscles. You probably know now that it's a muscle memory thing. You don't even have to think about how you're going to write out the letter A at this point, even if it's uppercase versus and not lowercase. Large motor units are more for strength and control. We wouldn't take a motor unit out of your gastrocnemius in your leg and try to get it to write a letter A, because it doesn't have the fine degree of control. But it's got a lot of strength in it. So it produces powerful contractions that are generated by large motor units with hundreds of fibers. Gastrocnemius has about 1,000 muscle fibers per neuron. So each motor unit can have at least 1,000 muscle fibers. In your eye or your hand, each motor unit is about 3 to 6 muscle fibers. The neuromuscular junction is the synapse. You remember we talked about synapses in chapter 12. These are synapses here. Same thing, whenever a nerve fiber is in target with its when it has connected with its target cell, that's a synapse. So the neuromuscular junction is a synapse. Between the neuron and the muscle fiber. Each terminal branch, remember that it can separate out. Remember the axon terminal has this branching like network of, if you want to look at it like a tree, the top of the tree is the dendroid and the cell body. The bottom of the tree are the roots, which in this case are the axon terminals. Each terminal branch of the nerve fiber within the neuromuscular junction forms a separate synapse with the muscle fiber. So one nerve fiber can stimulate muscle fibers at several points within a neuromuscular junction. With looking at the neuromuscular junction in a little bit more detail. So we're looking at one of these right here. One of these right here. One of these right here. Here we only see 3 synapses, but we can see there can be several of them. So the synaptic knob is the swollen end of the nerve fiber. It contains the synaptic vesicles that are filled with acetylcholine. A synaptic cleft has a gap between the is a gap between the synaptic knob and the sarcolemma. Here's your synaptic knob. Here's your sarcolemma. The motor end plate is the region of the sarcolemma opposite the synaptic end, which contains the acetylcholine receptors. So here you'll see the blue here is the acetylcholine receptors. The Schwann cells, remember what are the Schwann cells? Do you remember? What were Schwann cells? What were Schwann cells? Rachel, I can see that you're muting and unmuting. Do you know what, do you have an idea? Quentin, what are Schwann cells?
So I didn't know, so I just looked it up for the class or whatever, and it says they are any of the cells in the peripheral nervous system that produce the myelin sheath around the neural axons.
Exactly. So those are the neural glia that produce the myelin in the peripheral nervous system. So they also are involved in the envelope, this whole entire thing. We don't have them here, but they're going to envelope this whole entire thing and surround it. Synapses get stronger and stronger as the myelin around them gets better and better at enveloping them. The synapse gets made and then it gets strengthened by increased myelination around it. It's a lot of how learning works too. But it's rather than have it be the myelin in that case, it's the oligodendrocytes. Because that's happening in the central nervous system. So acetylcholine, an action potential, arrives at the synaptic knob. The acetylcholine is released from the synaptic vesicle. It enters into the synaptic cleft. The acetylcholine then binds to its acetylcholine receptors. And then a muscle action potential is then initiated. One thing that we don't mention here that I think will get mentioned in a moment here is that calcium is actually involved also, because calcium is what allows for the acetylcholine to be released. So the action potential comes down the axon. The voltage change results in the opening of calcium channels here. Those calcium channels result in exocytosis of the neurotransmitter. The neurotransmitter moves through the synaptic cleft and binds to its receptors. Acetylcholine. I read in your book, I was doing a little bit of review last night and reading in your book a little bit. And one of the things that the book says is that every action potential results in about 60 calcium channels being opened up. Actually, wait. Every action potential results in about the exocytosis of about 60 synaptic vesicles of acetylcholine. And the result is that there's about 1,000 molecules of acetylcholine released into the synaptic cleft. So every action potential, 60 vesicles released into the synaptic cleft. And 60 vesicles is about 1,000 molecules of acetylcholine. Skeletal muscle maintains a more negative resting membrane potential. So keep in mind that muscle here can, just like neurons, is capable of firing an action potential. Muscle maintains a more negative resting membrane potential. There are more anions inside than outside, just like there was in the neuron. Just like there was in the neuron, there's an excess of sodium in the extracellular fluid and an excess of potassium in the intracellular fluid. The T-tubules membranes also contain chloride channels, which makes the cell more permeable to chloride, along with potassium leak channels. The resting membrane potential, just like it was within the neuron, is maintained by the sodium-potassium pump. And potassium leak channels. Neuromuscular disorder, myasthenia gravis, is an autoimmune disorder. So this is where your body is attacking itself. Your immune system is all of a sudden recognizing that your own cells are foreign and attacking attacking those cells. That's the basis of all autoimmune disorders. Is your immune system all of a sudden recognizes that your own cells are foreign. Antibodies target the acetylcholine receptors. So antibodies that your body produces targets your own acetylcholine receptors on skeletal muscles. The acetylcholine receptors are destroyed, and as a result, when the acetylcholine is released from the synaptic cleft, there aren't very many receptors for it to bind to on the muscle because they've been destroyed. The result is that we have a slower neurotransmission. The muscle is less likely to fire an action potential. There isn't as acetylcholine just doesn't bind, and so there's the muscle potential, action potential in muscles become decreased. The result is that you have muscle weakness. We have certain drugs like botulinum toxin, which blocks the release of acetylcholine from synaptic vesicles. So this would be like a neuromodulator, if you remember that word from chapter 12. It's a neuromodulator. It can be found in improperly canned foods. Tiny amount can cause death by paralyzing the respiratory muscles. It's also used as the medicine Botox. Can help with a crossed eye strabismus, treat strabismus. Blepharospasm, uncontrollable blinking, spasms of the vocal cords that can interfere with speech, and be used as a cosmetic treatment to relax facial muscles that could cause facial wrinkles. I've decided I don't ever want to have a needle injected into my face intentionally, but we won't say anything. Questions? Phases of muscle contraction. That we have an excitation, then we have an excitation contraction coupling, where the excitation is actually leads to the contractions. So excitation is a process in which muscle action potentials lead to, nerve action potentials lead to muscle action potentials. Excitation contraction coupling are events that link the action potentials on the sarcolemma to the activation of the myofilaments, preparing them to contract. Contraction is a step in which the muscle fiber develops tension and may shorten. Laxation is when the stimulus ends and a muscle fiber relaxes and returns to its resting length. So excitation, action potential arrives at the synaptic knob, aka a nerve signal arrives at the synaptic knob, and it opens the voltage-gated calcium channels in the synaptic knob. The calcium enters and stimulates the release of acetylcholine from the synaptic vesicles into the synaptic cleft. Acetylcholine diffuses across the cleft, and two acetylcholine molecules bind to each receptor and open the receptor, open the channel. So you bind to the receptor and open the channel. Sodium enters and shifts the resting membrane potential from a minus 90 millivolts to a positive 75 millivolts. Potassium exits and returns the resting membrane potential to minus 90. The quick voltage shift is called an end membrane plate potential. Bear with me here. Called an end plate potential. Sodium enters, potassium then leaves. So the voltage change in the end plate region opens nearby voltage gated channels, producing a muscle action potential or an end plate potential that spreads over the sarcolemma and travels down the T tubules. Remember, those are deep indentations into the sarcolemma. And travels along. So the action spreads down the T tubules, opens voltage gated ion channels in the T tubules and calcium channels in the sarcoplasmic reticulum. This is where that ryanodine receptor is found that we were talking about earlier in the case study. Calcium leaves the sarcoplasmic reticulum and enters the sarcoplasm. So that's the excitation. The spreading of the action potentials down the T tubules and the opening of the channels in the sarcoplasmic reticulum to release the calcium. Calcium binds to troponin on the thin actin filaments, and the troponin-tropomyosin complex changes in shape and exposes the active sites on actin, allowing myosin to then bind. At that point, we have this cross-bridge cycling, the sliding filament model, where there's repeated attachment of actin and myosin. Every head where myosin can bind actin has an ATP binding site and has an enzyme at that same spot that can hydrolyze ATP. So the myosin heads hydrolyze ATP and become energized. This is what we call recovery stroke, where we're cocking, just like you're cocking a gun. You're cocking the myosin head. When they're energized, the myosin heads bind to actin, forming cross bridges. Remember that calcium is allowed for these active sites on actin to be activated or be exposed to myosin, and allowing myosin to bind. The myosin cross bridges rotate toward the center of the sarcomere. They grab a hold of it and push it forward. The actin, they grab a hold of the actin and push it forward. Grab a hold of it and push it forward. Shove it to the left. In what we call power stroke. At that point, they release the ADP. Adenosine diphosphate. While still attached to the actin, they bind another ATP. And once they've bound the ATP, the myosin can detach from the actin and attach to the next binding site a little further down on the actin. And this cycle of calcium ATP hydrolyzation, myosin binding, Myosin pushing the actin forward through this power stroke, releasing, having another ATP bound hydrolyzation, and then release again. Happens over and over again as long as ATP is available. Each of the 600 cross bridges in one thick filament can attach and detach approximately 5 times per second. So myosin pulls on the actin, causing the thin filament to slide inward. And as a result, the Z discs move toward each other and pull on neighboring sarcomeres, which results in a shortening of the myofibril. Thanks to the structural proteins, there is a transmission of force throughout the entire muscle, resulting in the whole muscle contracting. One thing to keep in mind here is the Z discs are being, the Z discs are being pulled together because the thin and thick filaments are getting more and more overlapped as myosin moves actin over the top of themselves. So the lengths of the thick and thin filaments don't change. It's their overlap that changes. So from being this to this to this. Nerve stimulation and acetylcholine release will then stop. And acetylcholine esterase breaks down the acetylcholine and the fragments are reabsorbed into the synaptic knob. Calcium is pumped back into the sarcoplasmic reticulum by active transport. And the calcium binds to calsequestrin while being stored in the sarcoplasmic reticulum. The calcium pumped back, tropomyosin reblocks active sites on actin, and the muscle fiber ceases to produce or maintain tension. And the muscle fiber then returns to its resting length. So just as an overview and review of this, nerve impulse from excitation to relaxation. Nerve impulse arrives at the axon terminal of the motor neuron and triggers the release of acetylcholine. Acetylcholine diffuses across the cleft and binds to the receptors in the motor end plate and triggers an muscle action potential. Acetylcholine esterase in the synaptic cleft destroys acetylcholine, so another muscle action potential can't arise unless more acetylcholine is released. Once the muscle action potential has been initiated, it starts traveling along the T tubules, opening calcium release channels in the sarcoplasmic reticulum, which allows for calcium ions to flood the sarcoplasmic reticulum. The calcium binds to troponin on the thin filament exposing the binding site for myosin. Power strokes using ATP involve where the myosin heads bind to actin and swivel and release, pulling the thin filaments closer to the center of the M line. That would be right here. When the muscle relaxes, the calcium release channels in the sarcoplasmic reticulum close. The calcium active transport sites use ATP to restore the low levels of calcium ions in the sarcoplasmic reticulum. The troponin tropomyosin complex slides back into position where it blocks the myosin binding sites on the actin, and then muscle relaxes. Are there questions? Let's take a 10-minute moment here. We'll come back at 10:59. You have questions, let me know too. I ask you if you have questions and then I don't let you respond. Let me know if you have questions. Sometimes that's not so so there's some truth to what I'm saying here. This is also the cost for So I wanted to make a comment, and if you're here, if you're not here, you just won't hear this story, which is okay too. So I made that comment I always think at the end of the semester, if everybody makes it and nobody dies, it's a success. But that's not so funny, at least to one of the students in this class, and I know that. But part of the reason when I make that joke, part of it is not so funny. And the reason is one of my first years I ever taught, started teaching college students. I was at Idaho State University and I was a teaching assistant for Idaho State University. And that semester, I actually had a student commit suicide. And so part of that is meant to be a joke and part of it is not. Because that was probably one of the more trying experiences I've ever had as a professor. So it's funny, but it's not at the same time. I wanted to try to pull up a video. And I'm not sure if it's going to happen fast enough. Why this is taking so long. There we go, maybe. There. Oh, not that one. Go back. This is I want to really quickly, there's 2 videos here. I'll post both of these, but let's watch this first one. It's just 3 minutes in length. And then I'll transfer this over after class.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The 2 myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
I will, so I have that under a different class, but I will put it under our class today. That video, I'll post it under this week's power module. So following death, rigor mortis, following death, what it essentially is the breakdown of the sarcolemma and the covering or the membrane that encloses the sarcoplasmic reticulum, resulting in higher levels of calcium within the cell. What happens when that happens? Well, troponin binds to calcium, which exposes the active sites of myosin, resulting in myosin binding to the actin. However, there's not more ATP being produced. And so the myosin is stuck, bound to the actin. So their ATP is needed to bind to the myosin heads and allow for the cross bridges to detach. But since ATP synthesis and other cellular processes quickly cease following death, this keeps the actin and myosin filaments linked together. And the muscle will remain rigid until the myofilaments start to decay. Questions? That is by far probably the most complicated part of this chapter. So congratulate yourself if we've made it through this far. Length tension relationship. The length tension property of the whole muscle is a relationship between the muscle length. Remember that the filaments don't change length. It's the overlap that's changing here. So the muscle, this is that length tension relationship, where it's a relationship between the muscle length and the force that the muscle can produce at that any given length of muscle. If the muscle is too overlapped at the point that a contraction is initiated, there's not any more overlap that can occur. And so the muscle doesn't contract very well if the muscle is too short in length. No more overlap can occur, so a contraction is not gonna, an excitation of the muscle resulting in contraction isn't gonna have a big effect because all of the overlap is already there. When there's partial overlap, that's the resting and optimal operating length. When the muscle is not overlapped at all, the thin and thick filaments, then even if a muscle contraction occurs, the actin doesn't have a spot that it can actually grab onto and pull the myosin forward. If it can't grab onto a spot, then there's not going to be a lot of contraction either. So the optimal length is when there's partial overlap, but not too much and not too little between the thin and thick filaments. As it turns out, the natural resting length of our skeletal muscles maximize the ability of muscle to contract when stimulated, where they're always partially overlapped already. This resting one, partially overlapped already. The nervous system helps maintain a certain amount of muscle tone, partial contraction, to ensure the resting muscles are near their optimal length so that they can get excited, the excitation can occur and contraction can start. If the muscle is too stretched before stimulated, a weak contraction results. And there's a minimal overlap between the thin and thick filaments, which results in only minimal cross bridge formation. Only minimal overlap. To generate most muscle force, sprinters don't start a race standing up. They start partially contracted. Gastrocnemius is already partially contracted if they start in the right position. If overly shortened before stimulated, a weak contraction results. And the thick filaments will just, yes, there's a contraction, but it's weak because the thick filaments are already butting up against the Z disc. And there's no more distance to shorten. No more length to actually reduce. You can't get any shorter. You can't generate any more tension. If you can't get any shorter, you can't generate any more tension. Questions? Myogram is defined as a chart of the timing and the strength of muscle contractions. The threshold is a minimal voltage necessary to generate an action potential in the muscle fiber and produce a contraction. So just like we saw in the neurons, there's a minimal voltage necessary to initiate an action potential. Same thing here, there's a minimal voltage necessary to produce a contraction. It has, and then at that point, it's an all or nothing. A twitch is a brief single contraction of all the muscle fibers in a motor unit in response to a single action potential that reaches threshold. So it's threshold, got to reach threshold. And once you reach threshold, there's a contraction. And a single contraction is called a twitch. If there's only subthreshold stimuli, there's going to be no contraction. At threshold intensity or above, a twitch is going to be produced. Stages of muscle contraction. So initially there's a latent period. This latent period is that brief time of where an action potential is reached at the synaptic knob. It releases acetylcholine into the cleft. The acetylcholine binds to the receptor and initiates the muscle action potential, which results in more calcium being released into the sarcoplasm, and which then binds to the troponin, which then shifts the troponin tropomyosin complex off the active sites of actin. So all of this is occurring during the latent period. So this is a time required for excitation, contraction, and tensing of the elastic components to occur. The action potential, muscle action potential propagates over the sarcolemma, remember that's plasma membrane, and the calcium is released from the sarcoplasmic reticulum. Calcium then binds troponin. The contraction phase is the time when the muscle generates an external tension. This is the force generated to overcome the load and cause movement. The tropomyosin moves and the myosin binding sites are exposed, and cross-bridge cycling occurs. And the sarcomeres then shorten.
So I don't know if something's happened. And when she called me directly and said she was sick, and I anyway.
During the relaxation phase is a time when the tension declines to baseline. The sarcoplasmic reticulum reabsorbs the calcium. The myosin heads detach from the actin and tension decreases. The tropomyosin covers the actin active sites. And this whole phase is going to take longer than the contraction phase. Even if the same voltage is delivered, different stimuli cause twitches varying in strength because of what?
But I don't know if you know, with the family. Yeah. I suspect you know. Yeah. And I just don't. Unsettled that. Or psychological. And function stuff. To adjust your medications. And I thought, oh, yeah, you can do that. You know, so
Any answers?
apparently they took the imbalance, which I think. So I don't know if this risk.
So really quiet. I would say that a lot of these could be factors, but we do know that the muscle starting length influences tension generation. How much overlap is already there? Remember, going back to the length contraction relationship or length tension relationship. Muscles must contract with varying strengths for different tasks. Muscles are stimulated, stimulating a nerve with higher voltages or stimulating a muscle with higher voltages will produce a stronger contraction. I think this is a typo right here. Stimulating the muscle with higher voltages will produce stronger contractions. Because higher voltages excite more nerve fibers, which stimulate more motor fibers to contract. Different fibers are going to have different threshold levels. Some have lower thresholds, some have higher thresholds. The lower ones with lower thresholds will be activated first. The ones with higher thresholds will be activated next. The strength of a muscle contraction depends on the strength of the stimulus. Showing a myogram here, remember that's the muscle strength. A 0 to 0.04 action potential or threshold show no observable contraction. This is meaning it's a subthreshold stimuli. A muscle increases its response from the threshold stimulus up to the maximal stimulus. So as the voltages get higher and higher, the muscle will increase its response until it reaches the maximum. Any stimulus above that maximal stimulus, because we've already reached the max, will elicit the same maximal response. The strength of a muscle contraction depends on the frequency of the stimuli also. So it's the strength of the stimulus and then the frequency of the stimulus. Low frequency stimuli are going to produce identical twitches. High frequency stimuli are going to produce a temporal summation. Or a wave summation where one stimuli will be added on the last one because they're coming at a higher frequency. Remember how we got summation in nerve impulses in chapter 12? We get them here too, and with muscle contractions. So its muscle fiber is re-stimulated before it's completely relaxed. So the second twitch is added to the first one.
And I'd always figured you don't know what they're like.
Only partially relaxation is going to occur between the stimuli. And this is called incomplete tetanus. When you can't pump all the sarcoplasmic, all the calcium back into the sarcoplasmic reticulum between the two stimuli, some of the calcium is still out in the sarcoplasm, activating the troponin when the next action potential comes in. Is this clear?
Yes.
So stimulation at a higher rate results in sustained muscle contraction that permits partial relaxation between the stimuli. This is unfused tetanus. On a naturally high stimulus, in lab experiments, you don't get this outside of a laboratory, resulting complete or fused tetanus. We can get this if we isolate a gastrocnemius muscle of a frog and we fire a bunch of action potentials into it.
My son and daughter-in-law said, we're not doing
But we don't get it all. We don't get it without an unnaturally strong stimulus. So the strength also of the motor contraction, so it depends on the strength of the stimulus, it depends on the frequency of the stimulus. It also depends on the size and number of motor units involved. Larger motor units generate more tension due to larger numbers of muscle fibers contracting. As a greater number of motor units are activated, the muscle can generate more tension to lift heavier loads. All of the muscle fibers in one muscle unit contract in unison.
and what she said is all.
Muscle fibers of a motor unit are dispersed throughout the muscle and aren't clustered together. As a heavier load requires more motor units to be involved, we can recruit more motor units. Motor recruitment is where more active motor units are involved. The weakest motor units are recruited first, followed by the stronger motor units.
Well, I said to her, I said, what did I tell you? And she said, brownies. I mean,
All right, you guys, my mom is talking in the room and I'm getting really easily mentally distracted with it. I'm gonna walk this into my another room. Bear with me just for a second. Bear with me. hope you're all still here with me, but I need a little bit of a change in venue so that I would not get distracted by what she was saying. So this is a process by which weakest motor units are recruited first, followed by stronger motor units. So if the load is smaller, you only recruit a few motor units. If the load is larger, you recruit more motor units. Come on in, Paisley. Push it a little more. Mom, come open my door, my bedroom door. As more motor units generate more contraction, additional motor units are activated. Paisley, don't let me. So as more weight is required to move a particular amount of mass, as more force is required to move a particular weight, more motor units are recruited, resulting in more contraction. If a muscle contains a limited number of motor units, then how do we grow up or lift weights? Do you remember what this was called when motor units gets bigger? Was the hypertrophy? Yep, good job, Aaron. Yes. Paisley, no. Evan, no, buddy. Okay. So muscles do not increase in the number of motor units. They don't increase in the number of muscle fibers in a motor unit. They only increase in the number of myofilaments. How do they increase in the number of myofilaments? Satellite cells. So you start lifting weights. How many? I like to lift free weights. How many of you? I know that I'm sure we have other people who like to lift free weights. After you're done lifting free weights, your muscles are a little sore sometimes. What you've done is you've done a partial breakdown of the muscle. And what happens is it recruits satellite cells to heal those injuries that you've made. And add more myofilaments. So the satellite cells fuse to the injured muscle and add more myofilaments. Because the myofilaments in the satellite cells adds to the myofilaments that were already there. So hypertrophy results in an increased diameter and larger fibers due to the addition of more thin and thick filaments. So maximum tension is dependent on the rate at which nerve impulses arrive, the amount of stretch that occurs before contraction, and the size of the motor unit. Well, how do muscle fibers meet their energy demands during rest and exercise? It's a nutrient and oxygen availability. What are all the functions of ATP in muscle contraction? Can you describe them? The first one is hydrolysis of ATP that on the myosin, which is needed to energize the myosin heads so it binds to the ATP. And then we need myosin to pump the calcium back into the sarcoplasmic reticulum. It's also required to restore the resting membrane potential. Remember that sodium potassium pump needs ATP also to pump the sodium and potassium gradients again, and so we get concentration gradients restored. Muscle contraction demands an enormous amount of ATP. A contracting muscle uses approximately 2 million ATP molecules every second. Without a constant supply of ATP, muscle contraction will cease. There's two main ways that we get ATP synthesis. We have aerobic respiration, which produces a lot of ATP and does not generate lactic acid. But aerobic respiration requires a continual supply of oxygen. Anaerobic fermentation enables the cells to produce ATP in the absence of oxygen, but it produces very little ATP and generates lactic acid. Aerobic respiration uses oxygen from the myoglobin. Remember, we talked about that at the beginning of this chapter, how it's a derivative of hemoglobin that binds to oxygen and stores it in muscles. Then a phosphagen system comes into place, which we'll talk about next. And then we have a glycogen lactic acid system that involves anaerobic fermentation. And then we have aerobic respiration kicks in again when the cardiopulmonary function has enough time to get into gear and provide the energy necessary. But that takes a little bit of time for it to occur. So initially it's from the stored myoglobin within the muscle. Next thing, it's this phosphagen system. Next is this glycogen lactic acid system. And then after about 40 seconds, aerobic respiration will kick into because it's at that point supported by the cardiopulmonary circulation. Takes a little while to get this up to gear. So in the first 8 to 10 seconds, oxygen is briefly supplied by myoglobin. Two enzyme systems also contribute here in what's called the phosphagen system. This system transfer different inorganic phosphate molecules to ADP to produce ATP that can be used for muscle contraction. So they're transferring the inorganic phosphate molecules from one structure to ADP. Two different systems. Myokinase transfers from one ADP to another, one phosphate to another ADP, converting the latter to ATP. Sorry, my cat keeps jumping over my computer here as I'm sitting on the floor. Creatinine kinase obtains inorganic phosphate from creatinine phosphate and gives it to ADP to make more ATP. So first one transferring ADP, making ADP into AMP, giving that phosphate to another ADP to make ATP. Remember, ADP is an adenosine residue with two phosphate groups. AMP is an adenosine group with one phosphate group. We talked about that in chapter 2. So you're giving up that phosphate of one ADP and giving it to another ADP to make ATP. That's the first way we can make ATP during this phosphagen system. The second one is creatinine kinase will get the phosphate group from a molecule called creatinine phosphates and give it to ATP. The combination of these two systems right here provides nearly all the energy for short bursts of activity. They're recharging the ADP that are available in the muscle to make more ATP. And no oxygen is needed for this system, which is a phosphagen system. As the muscle activity continues, the amounts of creatinine phosphate is depleted, and muscles then will obtain glucose from the blood and their own glycogen storage. Remember, glycogen is a stored glucose molecule. And because there's still not enough oxygen available, because the cardiopulmonary system isn't up to snuff and we've ran out of myoglobin having oxygen bound to it. In the absence of oxygen, glycolysis will generate 2 ATP for every glucose molecule and produce lactic acid. So this is in the next little while. Bear with me just for a second here. 1 to 3, what happens when I don't have my mouse? in the first 1.3 to 1.6 minutes. After about 40 seconds, the respiration cardiovascular systems will start to deliver oxygen fast enough for aerobic respiration to meet the most of the muscle's ATP demand. So initially it's aerobic respiration from myoglobin, and then creatinine phosphate, and transferring inorganic phosphate groups from one ADP to another ADP to make ATP. And then it's anaerobic respiration, where we start breaking down the glucose storages within the muscle to produce ATP. And after that, the respiratory and cardiovascular systems are now where they need to be to start. They're above the limit. They're working hard enough that they can deliver the oxygen needed for aerobic respiration to begin again. When we produce aerobic respiration, we get 32 ATP per glucose molecule, but we have to have oxygen for that. When it's anaerobic, we only receive 2 ATP per glucose molecule. Once glucose molecule, the glycogen stores start to get depleted within the muscle. We're running out of glucose. Then we start burning the fatty acids to produce ATP. Is this clear?
Yes.
Muscle fatigue is an inadequate release of calcium from the sarcoplasmic reticulum. It's due to a lot of different factors. These are the different possibilities. Inadequate release of calcium, depletion of creatinine phosphate, and oxygen and nutrients like glycogen or fatty acids, the buildup of lactic acid and ADP, and an insufficient release of acetylcholine at the neuromuscular junction. Well, what if we start running out of acetylcholine in the synaptic knob? Why do you continue to breathe heavily for a period of time after the muscles stop, after you stop exercising? I'm realizing that the more fit I tend to get, the less I tend to breathe heavily afterwards. And if I go more than a couple of weeks without exercising, I try to take at least a time every week. I take about an hour and a half on Saturday nights, and it's my self-care time where I work out. And if I can just get that in every week, I tend to be a happier person, a little calmer person. And my muscles don't struggle from week to week to get up and get moving when I exercise like that. But why do you breathe so heavily after you stop? you have to meet an oxygen debt. You have to restore the oxygen debt. It's the difference between an elevated rate of oxygen consumption following exercise and your usual resting rate. You need some extra oxygen to pay back or restore the metabolic conditions to the resting state. This excess post-exercise oxygen consumption, or EPOC, is needed to aerobically replenish the ATP, some of which helps regenerate the creatinine phosphate stores, replace oxygen reserves on myoglobin, provide oxygen to the liver that's busy disposing of lactic acid, and provide the oxygen to many cells that have elevated metabolic rates after exercise. This consumption can be 6 times bigger than the basal consumption and last about an hour. To delay the onset of muscle fatigue, not all the motor units in a muscle will contract at the same time. Some will remain relaxed while others are contracting. If the contraction continues, greater force is needed and motor units will be recruited. True or false? Genesis, what do you think?
I was thinking true, but I'm not completely sure.
Why do you think it's true?
I don't know, it just makes sense.
Well, you're correct. Remember that they don't all contract at the same time. They take turns. And we can recruit more motor units if we need them. So Genesis, you're completely right. Did you know that some of your muscles are contracting even when you're at rest? Remember, you have the autonomic nervous system is involved in innervating some of your skeletal muscles. A skeletal muscle can exhibit a small amount of tension to stabilize positions of bones and joints. It's established involuntarily by neurons in the brain and spinal cord. And this is part of your cerebellum's job and the reticular activating system. Small groups of motor units are alternatively active and inactive in a constantly shifting pattern to keep muscle firm. skeletal muscle fibers can also vary in the amount of myoglobin they contain. Remember, this is the first oxygen store. Remember, this is the first 8 to 10 seconds of exercise where we get the oxygen we need for ATP stores is in the myoglobin. The first 8 to 10 seconds. Keep that in mind. First 8 to 10 seconds, this is where we get it from. Sorry, as my dog shifts my computer too. Bear with me. Myoglobin is higher in dark meat or muscles that have more capillaries. Like your legs. Your back muscles don't contain as much myoglobin. Your legs tend to contain a lot more myoglobin. If you were looking at the turkey this last year, breast meat, more white meat, less myoglobin, less capillaries. Legs and wings, more dark meat. Depending on the type of bird. Turkeys, their wings don't contain as much dark meat. Why do they not contain as much dark meat? Because they don't fly. They don't really use those wings as much as a duck would. Ducks, their wings contain more dark meat. More myoglobin because they use them to fly. Your sparrows tend to have more myoglobin in their wings because they use them. Ostriches don't have any myoglobin in those muscles. So this can change. Also, the levels of myoglobin can change if you need it. They can also vary in the muscle fibers can also vary in the diameter of fiber, contraction velocity, and the production by which we make ATP. The method by which we make ATP. And resistance to fatigue can vary. Muscle fibers can be classified into 3 different types. Slow oxidative, and the slow versus intermediate oxidative glycolytic, and fast. The slow, intermediate, and fast is based on how quickly the myosin hydrolyzes the ATP. How quickly do they break down the ATP so that they can make another cross bridge with another active site on actin? In the slow fibers, it's slower. In the fast fiber, in the intermediate fibers, it's somewhere in the middle. In the fast glycolytic fibers, it occurs a lot faster. The oxidative versus glycolytic is where do those fibers tend to get their ATP? Do they tend to get oxidative, which means oxygen, or glycolytically, meaning they anaerobically? the proportions of oxidative and fast glycolytic fibers can vary, and they can change with exercise training. Marathon runners have about 82% slow oxidative fibers in their quadriceps femoris muscle, which we'll talk about specifically where that muscle is, but it's the one right on the front of the thigh. Talk about that in lab. And 18% fast glycolytic in that same muscle. Swimmers have about 74% fast oxidative fibers in that muscle and 26% fast glycolytic. Average males 45 and 55. Sprinters and jumpers. Sprinters tend to have about 63% of fast glycolytic. They need that fast. That's why it's fast glycolytic. They need that push fast. Swimmers and runners need it for a longer period. It's more duration, it's not fast. If you think about your people who do this jumping and the sprinting, they need it fast. They're not looking for endurance, they're needing it for fast. That's the difference between those two, and you can change the fiber percentages. Some of it is also genetic. How many fiber, how quickly you can change back and forth between the two fibers and how quickly they can vary. Last part of this chapter, talking about smooth muscle cells. Smooth muscle cells contain thin and thick filaments not arranged in sarcomeres. They have what are called dense bodies that function as Z disks, though. Here's their striations. Bear with me just for a second. I'm gonna zoom in just a little bit. And I know that maybe this wasn't a good idea, but I'm gonna try it anyway. So here's the striations. Here's the thin filaments. Here's the thick filaments. Here's the overlapping. You still have it. But rather than having Z disks, you have these dense bodies. When the muscle contracts, the dense bodies get closer. They get closer together. So smooth muscle, usually activated involuntarily. Action potentials are spread through fibers via gap junctions. Same thing in cardiac muscle. The fibers are stimulated by certain neurotransmitters, hormones, or autorhythmic signals, where the muscle itself maintains rhythmic contractions. Each smooth muscle is like one giant sarcomere. Smooth muscle is found in the walls of arteries and veins, hollow organs, and the airways to the lungs, such as the bronchioles. These are also muscles that attach to hair follicles. These are what give you goosebumps. And adjust the pupil diameter and adjust the focus of the lens in the eye. So these can be innervated by the oculomotor nerve. Remember, that's what's involved in the pupil diameter. And adjusting the lens. Smooth muscle contractions start more slowly and last longer than skeletal cardiac muscle contractions, but they remain contracted for longer too. Oh, it says that. So start slower and last longer. Smooth muscle can shorten and stretch to a greater extent than skeletal cardiac muscle. Smooth muscle also lacks striations. And sarcomeres. And you guys, that is actually the end. Do you have questions for me? Is that a nope?
No.
Okay. How often do I actually let you go early? Not very often. But I will see you on Thursday. And like I said, Lakeisha, I just saw your email and I'm saying yes to whatever it was.
Okay.
I didn't read it. I just saw it. And
All right. Thank you.
And Quentin, I'm saying yes to yours too.
All right. Thank you.
All right. You guys have a wonderful 2 days. I will see you on Thursday.
All right. Bye.
Thank you.
You
See
then.
ya.
Thank you.
Okay, so this is not relating to the class, but
Okay.
I was trying to get into D2L and like none of my courses will load up. So could you do me a favor and check to see if your D2L is working?
It
Just
might
so I
not be
can-
working for is it for next semester or this semester?
This semester, like none of my courses, like it will log me into the page, but then none of my actual like courses will pop up. Like not, it just keeps loading. So I just want to see if it's like just my computer
My guess.
or if it's actually D2L that's like messed up.
I was having those problems too.
Really?
The tablet that I was using died, and then I tried to log in on my laptop, and it took like 10 minutes for the course to show up, and it actually did that the other day too. So it's something I don't know, but it happened to me too.
Yeah, this is the first time this has ever happened. And it's been sitting loading in the background for like probably the past 10 minutes of class, just because I was like getting ready to do more schoolwork. And I was like, why isn't it loading?
All right, Genesis, you're gonna make me cry because I have a class in 20 minutes.
Yeah, it might
I
not
have
work out.
homework due.
I don't think it's just you.
Okay, I just wanted to make sure it wasn't just me.
Oh, this is really gonna stink.
Yeah,
Um,
last time this happened, I went to a different college before this. And
Yeah,
last time this happened, they had to like cancel classes all day because D2L crashed.
are you serious? This isn't good at the end of the semester.
Yeah, definitely not, because I know a lot of people have a lot of work due right now.
Well, and professors, if you saw, we kind of we've squished things together a little bit this semester.
Yeah, all of my
Thank you.
professors have done it right towards the end, they're like, oh, we have to do this, and that.
All right. Well, I'm glad I'm not the only one who feels this way. But let's, oh, shoot. Nope, Genesis, it's not just you.
Okay. Thanks.
I'm glad we at least got it loaded at the beginning of the class.
Yes.
All right. All right. I will see you later, but it's not just you.
All right. Thank you. Have a good
Okay.
day.
All right.
Thanks. Bye.
Bye-bye.
I want to go out.
I know this isn't Daisha.
It's
It's some-
Jamie.
Jamie,
Yeah.
can I do it? You got a question for me?
Oh no, I have to respond to your email. I need a lot of my things opened up, and I thought you could just like hit a button and open everything that
No.
was closed.
No,
but
I
you
can't.
Yeah, you need a list of it. So
Yes.
how's it easiest to tell you, because it's so much?
Just email it to me, because right now, the fact that I can't load anything, I need a list on Word. Send me a Word document. Tell me
Okay.
when you'll have it done, approximately. I have to have my grades in by about the 17th. So you've only got about 10 days.
Yeah, I need to get cracking. Okay. And then I guess that's it then. I'll figure it all out. And I mean, some of the stuff is open. I was doing some of it last night too. So I'll figure it out for sure what I need you to open, and then I'll
Okay.
send you an email.
All right, Jamie, first, do me a favor, do it in this order. Worry about the exams.
All of the exams.
They carry the most weight.
Okay.
Then worry about the exams and the lab quests. You're gonna have one more lab quest. Worry about those.
Okay.
Exams, then lab quests.
Yeah, actually the lab quests I definitely need opened. All of those are closed.
Okay, well, I don't think, oh wait, I haven't even, I will try to do it. I only have a few minutes and I have to figure out why my computer isn't working. So
Okay.
I probably won't get it done for at least an hour. I may
Okay.
still email me and remind me. 'Cause
Okay,
this
thank
is
you.
gonna be a busy day for me. So email me and remind me.
Okay, thanks. I hope it all works out.
You and me both. I'll talk to you later, okay?
Okay, bye.
Thank you. Bye-bye. Courtney, are you there?